Hi folks, welcome to my channel, The Average Dermadoc. Let's begin the show. Steps of collagen biosynthesis. Before moving to our topic, first we will see what is collagen. It is the main structural protein forming fibrous connective tissue of dermis along with elastin and reticulin. It contributes to 70 to 80% of the dry weight of the skin. We'll decode now each and every word. What is collagen? It is basically a protein, but structurally it resembles a fiber. That's why we are calling it as fibrous connective tissue. It forms the main connective tissue in the extracellular matrix along with other connective tissues like elastin and reticulin. Reticulin is nothing but a type 3 collagen only, but it is categorized separately. Why? We will see now. Our collagen is the main part contributing to the connective tissue. How much it contributes? That is 70 to 80% of the dry weight of the skin. What is meant by dry weight? If we remove all the water content from the skin and weigh the skin, 70 to 80% of the weight is because of collagen. That much huge amount of collagen will be present in dermis. Clear? A protein, but which looks like a fiber. Coming to components of dermis. Now we have seen collagen, elastin, reticulin. Along with this, what and all structures are present in dermis, we will see now. First, cells. We all know that dermis contains cells and vascularity, that is blood vessels. Many cells like fibroblasts, macrophages, mast cells, but here the topic of important is fibroblast, because the main cell which produces extracellular matrix is fibroblast. Now fibroblast is a cell which is a living thing which produces non-living protein, that is contributing to extracellular matrix, that is collagen, elastin, reticulin, and also ground substance. Now simple thing. A cell, fibroblast, which is living, produces non-living extracellular matrix. Now we will see what is extracellular matrix. It contains fibrous connective tissue as well as ground substance. First we will see what are present in ground substance. Glycosaminoglycans, that is main glycosaminoglycan is hyaluronic acid. Proteoglycans, glycoproteins, water and electrolytes. Remember that entire dermis is an amorphous gel. Because of this water content and electrolyte content, this ground substance is forming a gel, amorphous gel. Within this gel, our collagen, elastin, reticulin, all this fibrous connective tissue will be embedded along with our cells like fibroblasts, macrophages, mast cells. All will be present inside this amorphous gel. Just like we are living beings, we build houses which are non-living. Both of them are embedded on the ground. Same here also. Cells, fibrous connective tissue, everything will be embedded in our amorphous gel-like substance, that is ground substance. In that, what is the importance of this glycosaminoglycan hyaluronic acid? We must have heard about fillers. If we use hyaluronic acid filler and fill the ground substance in old age, which will be lost, it will bring back the youth and support to the skin, volume of the skin. If we inject hyaluronic acid filler, it will bring back the youth and volume to the face. That is the importance of this ground substance, that is amorphous gel. In hypothyroidism, we must have heard the word mixed edema. There, these glycosaminoglycans will be increased, leading to puffiness of the whole body. Now coming to our topic proper, fibrous connective tissue. What and all present in fibrous connective tissue? Collagen, elastin, reticulin. We will see what is collagen. Collagens form thick bundles. First, fibrils will form, collagen fibrils, which arrange together to form fibers. Then all the fibers will arrange together to form huge thick bundles. these thick bundles provide tensile strength to the skin. If we forcefully give any mechanical pressure, we will tolerate that because of the tensile strength of the collagen, because of these thick bundles which gives strength and support to our skin. Clear? Fibril is just a thin exam paper thread, like we will tie after our exam with a thread. No, that simple thinnest thread is fibril. Then many such fibrils together will form a thick thread, that is fiber. Even more thickest one is bundle. Many such fibers will form bundle, that is our collagen. Coming to elastin, exactly opposite to this thin branching fibers. Fibers won't together form bundle. Instead, they will branch and form branching fibers, which are thin. Elastin, thin. Here it is forming bundle because it has to give strength, but elastin is forming branches because it has to give the property of stretching and recoiling, that is elasticity. If we pull the skin, it will go back to its normal position because of this elasticity. We can stretch the skin, and if we leave, it will go into its natural position. Remember a tree which is having a thin branch. If we pull and leave, that branch will go into its position immediately back because it is thin. That is elasticity, elastin. Suppose if we pull the same thickest branch which is very hard, if we pull, it won't come. If we pull even more harder, it will break and come to our hand. That is because strength. It is for strength, not for stretching and recoiling. So now, what if we lost our collagen? Our tensile strength and support will be lost, which will make skin fragile. We can easily extend our skin. Hyperextensibility. Collagen will provide support to blood vessels also. Now blood vessels will also become fragile. Easy bruisability, hyperextensibility, weak skin. Skin will become very weak. No strength and support. What if elasticity is lost? If we pull the skin and leave, no recoiling capacity. So it will form wrinkles. Old age people will have wrinkles naturally because of loss of elasticity. Now what is reticulin? As I have told you, it is also a collagen, type 3 collagen. Remember like this, it is type 3 collagen which is in the form of a net. That's why it is named reticulin, because of this network-like pattern, thin net-like pattern. It will be present around blood vessels, supporting blood vessels and internal organs like uterus, GIT. To support there, it will form a net-like structure. Not only because of this, it is having a separate staining pattern also. Here we can see collagen staining in routine stain, H&D stain, pink color. But reticulin is staining black with different stain, a special stain, that is silver. It is argyrophilic. On silver staining only we can appreciate black net-like fibers, that is type 3 collagen. Because of these differences from all the collagens, type 3 collagen is named as reticulin. Now we may get a question asking name 10 signs in dermatology, name 10 special stains in dermatology. Now we have seen one special stain, that is silver stain, which stains type 3 collagen into black. Another special stain is Verhoeven Van Giessen stain, which stains elastin black. If we stain with HNE, that is routine stain, not a special stain. Special stains are here. We have learned two. Verhoeven Van Giessen stain, which stains reticulin and which stains elastin black. Silver stain stains reticulin black. On light microscope, if we use HNE stain, we can appreciate collagen. If we use a silver stain, we can appreciate reticulin. If we use Verhoeven Van Giessen stain, we can appreciate thin branching fibers, that is elastin, in black color. So from this question itself, we have learned two special stains. All these fibrous connective tissues are present in outside the cell, that is extracellular matrix of the dermis. Clear? Dermis have cells, vessels, extracellular matrix, that is fibrous connective tissue and ground substance. Coming to types of collagen. Even if there is a slightest variation at the genetic level, there will be the resultant collagen formed will be different types because of genetic variability. Now we will see what are the different collagens present. Type 1 is present in dermis, particularly reticular dermis, very deep inside the dermis. Type 1, most abundant collagen is type 1. That's why I have star marked it. It is also present in bone. What if type 1 collagen is defective? It will obviously affect bone. Bone means osteogenesis imperfecta. Type 2 collagen, a very famous mnemonic, cartilage. It is present in cartilage. What if it becomes defective? Obviously cartilage will also becomes defective, chondrodysplasia. Type 3 collagen. What is type 3 collagen? Now only we saw net-like pattern, reticulin. It is also present in dermis, particularly upper dermis. Lowest dermis is type 1. Upper dermis, papillary dermis is type 3. And it also supports internal organs. I told you, uterus, GIT and surrounding the blood vessel. It also supports blood vessels. So obviously if it is defective, vascular type of Ehlers-Danlos syndrome will be the defective disease. Type 4, 7, 17, all these are related to basement membrane zone. If 4 is defective, it results in Alport's syndrome. Here we are talking about structural genetic defect. If antibody comes and destroys type 4 collagen, it leads to Goodpasture syndrome. That is different. Now here structural defect. If type 7 is defective, epidermolysis bullosa dystrophicalis. If type 17 is defective, epidermolysis bullosa junctionalis will be the resultant disease. Type 5 collagen is present in dermis. Same, whatever collagen is present in dermis, if it becomes defective, it will lead to Ehlers-Danlos syndrome. So everything related to dermis I have highlighted in green color. You can see all odd numbers, 1, 3 and 5. All these are present in dermis. Resultant disease may be, you can write Ehlers-Danlos syndrome. Various variants are there. You can write for 1, 3, 5. For 3, vascular type of Ehlers-Danlos syndrome. Clear? Applied aspects of collagen. There are many types of collagen. Out of those, I have mentioned very few. Now coming to our actual topic, biosynthesis of collagen, where it occurs not only in fibroblasts. Main thing is fibroblast only. F for fibroblast, it produces fibrous connective tissue and also a number of cells, smooth muscle cells, vascular endothelial cells, epidermal keratinocytes. Everywhere in the body it needs collagen, so many cells will produce collagen by themselves. Now we will see how collagen takes its birth, biosynthesis of collagen inside a fibroblast. Starting from genetic level, we will go to the degradation of collagen. First, we will have a basic idea of what is collagen. Collagen is a protein, that means millions of amino acids will together form proteins. Every third amino acid in that peptide chain will be glycine, which is a constant thing. That's why we have written glycine. What is X and Y? In 10th class, we have all studied max. Whatever we don't know, we'll take this as X, like that. Now here we have taken XY. Because it may be proline, it may be lysine, it may be hydroxyproline or hydroxylysine after hydroxylation. So it can be various combinations of glycine, proline, lysine, glycine, hydroxyproline, hydroxylysine. Like that, many variations may come, but constant amino acid is glycine. This is the basic structure of collagen. Now we will see how it is produced. First, from the nucleus, genetic level, DNA will transform to RNA, messenger RNA. We all know that it is transcription. Inside the nucleus, first there occurs transcription, that is DNA gets converted to messenger RNA. It contains the message and it is the future collagen. Now this mRNA enters the rough endoplasmic reticulum. where ribosomes will be attached and all the protein synthesis will happen there only. So now collagen is also a protein. Its mRNA has entered rough endoplasmic reticulum, where there occurs translation, that is mRNA to protein, that is translation. DNA to RNA, transcription, finished. RNA to protein, translation. Here it is not mature collagen protein, it is just a precursor. Alpha chains, pro-alpha chains, these are precursors of collagen. It is called preprocollagen. Pro-alpha chains are produced. Clear? After translation, we have got preprocollagen, which are alpha chains, pro-alpha chains. Now, after translation, two main post-translational modifications will happen. What are those? Hydroxylation and glycosylation. Total 3 steps will occur in rough endoplasmic reticulum: translation, post-translation modification. Hydroxylation is done by enzyme hydroxylases, which requires vitamin C, oxygen and iron. If vitamin C is defective, collagen synthesis will be obviously defective. If collagen is defective, blood vessels will become fragile, bleeding from the gums will occur, that is nothing but scurvy. Vitamin C defect will lead to scurvy because of defective hydroxylation. Why hydroxylation is that much important? Because it leads to formation of stable covalent crosslinks and stabilizes the triple helix configuration of collagen. Finally, collagen will attain triple helical structure. To attain that and stabilize that, we need hydroxylation. What happens in hydroxylation? We already know X and Y are proline and lysine. Proline hydroxylase converts proline to hydroxyproline. Same, lysine hydroxylase converts lysine to hydroxylysine. Finish hydroxylation. Next, glycosylation. Name itself suggesting us it will add some glucose and galactose. If it adds glucose, glucosyl transferase. If it is adding galactose, it is by galactosyl transferase. Clear? Glycosylation is by glucosyl transferase and galactosyl transferase, which adds glucose and galactose to mainly hydroxylysine, but it may also add to hydroxyproline also. Now what has completed now? Proline has converted to hydroxyproline and hydroxyproline has been added with glucose and galactose. So now we have got a glycoprotein. Collagen is actually a glycoprotein because of this glycosylation. Now we have got O-glycosidic linkage, that is glycosidic bonds, hydrogen bonds, disulfide bonds at the end of post-translational modification. We have got precursor molecules now, pro-alpha chains. 3 pro-alpha chains will together assemble into a trimeric molecule. 1 alpha chain, 2 alpha chain, 3 alpha chain. Total 3 alpha chains now arranged in a trimeric molecule. And they will intervene around each other via intra and interchain hydrogen and disulfide bonds. These alpha chains will intervene around each other 1, 2, 3 via intrachain bonds and interchain bonds, hydrogen bonds, disulfide bonds. So ultimately we have got a trimeric molecule of alpha chain which is intervening around each other, forming a triple helical structure. This is the final triple helix we have got after post-translational modification. Why I am drawing and showing you? In exam you have to draw this structure, triple helical structure, how it is formed because of disulfide bonds, hydrogen bonds, because of post-translational modification. Finally, what we got? Triple helical structure of preprocollagen. Now it is no more a precursor of collagen, it is a procollagen. Once we get the triple helical structure, now onwards we are calling this as procollagen. First preprocollagen alpha chains. 3 such alpha chains. Now because of glycosylation, hydroxylation, it has arranged into procollagen triple helix. Exocytosis. Next step is exocytosis. Now procollagen is formed. It will enter Golgi apparatus. Golgi apparatus is a gift packing organelle. Here procollagen will be gift packed to send it to its target destination, that is extracellular space. Now it has to reach its target, that is extracellular space of the dermis. All procollagen molecules are packed into secretory vesicles and then exocytosed. It is just thrown out in the form of vesicles. All procollagen molecules which are in the form of triple helix. Now what happens in extracellular space? It will contribute its fibrous connective tissue stature. Now we will see what happens in extracellular space. First, it will undergo cleavage of its ends by procollagen peptidases. I have drawn you a triple helix model of procollagen, but at the ends it won't be any triple helix, it will be just a trimeric molecule. This ends will be cut down by procollagen peptidases. After cutting down, only the triple helix will be left. Many such triple helix will arrange themselves into a fibril, that is collagen fibril. See, cleavage, ends are cleaved by procollagen peptidases, results in formation of insoluble preprocollagen, which are monomers of collagen. Now we have seen procollagen gift packed and sent out. If any gift wrapper is extra left out, we will cut it. No, same like that. Procollagen peptidases outside will see the gift. If it is okay, it will leave. If it is not okay, that is ends are not in triple helix model, no, it will cut off and leave only triple helix insoluble preprocollagen. Now it is no more a procollagen, it is a preprocollagen, insoluble monomer of collagen. So what will happen? All the monomers together will polymerize, aggregate itself to form collagen fibrils. Ultimately we have got our exam thread like thin structure collagen fibril. These fibrils together arrange in a thick fibers like manner. These fibers will have to get stabilized now. Which will stabilize? Lysyl oxidase will stabilize by covalent cross-linking, by forming covalent bonds between preprocollagen monomers. Many such preprocollagen monomers will be cross-linked by covalent bonds by lysyl oxidase enzyme, which requires copper, ultimately stabilizing collagen fibrils. Once it gets stabilized, many such fibrils will arrange together to form, organize into fibers and then to bundles. Only if it forms bundle, it will give us tensile strength and support. Finish our collagen biosynthesis. First, from genetic level, DNA will convert to mRNA, that is transcription. Then it enters rough endoplasmic reticulum where it undergoes translation and post-translational modification. Now it has modified into procollagen, ready to gift pack and send to its destination. Golgi apparatus packs the gift and sends to outside. In extracellular space, it is cleaved perfectly, leaving only triple helix structure, that is preprocollagen, which is the monomer of collagen. Now simple, all the monomers together will form cross links by lysyl oxidase, forming first fibril, then organizing itself to fiber, then bundles. Collagen, whatever takes birth whichever comes onto this earth will have an expiry date. After expiry date finished, now collagen has to get degraded. It undergoes continuous yet slow degradation by collagenases. This collagenase is a family member of a big family called matrix metalloproteinase, which contains many such enzymes, not only collagenase. How we are practically seeing collagenase getting degraded means in urine continuously hydroxyproline will be excreted, which is a degradation product. Only when collagen gets degraded, it will be excreted through urine. So collagen will be formed slowly, continuously, and degraded slowly, continuously. In cancer patients, if stage goes to final stage and metastasis occurs, that cancer cells has to move all over the body, so it will release collagenases and increases the destruction of collagen from the body. That's why cancer patients at end stages will become very, very thin. This is the story of collagen synthesis. Remember the channel name. Don't forget to like, share, and subscribe. Thank you. Hasta la vista. До свидания.